John Novembre. So John has had a very successful career studying population genetics. And one of the topics that John has been particularly very interested in studying is what evolutionary processes are driving the geographical differences in patterns of genetic diversity. And this is a particular topic that John has been working on since about 15 years ago, when he did his first studies studying the geographical spread of a mutation that gives HIV resistance when he was a grad student with Montgomery Slatkin. And then after that, he continued working on this particular topic, and he went to study how you can analyze patterns of genetic structure using principal component analysis. And he has developed more sophisticated tools recently to study patterns of migration between in a particular geographical area using patterns of genetic diversity. And he's going to touch on these topics today, and we are very happy to have him here today. So I will let John take it away.
Great. Thank you, Diego. Muchísimas gracias a todos. Es un placer estar aquí con ustedes. Voy a dar la charla en inglés, pero si quieren preguntar después en español está bien. So yeah, it's really a pleasure to be here, and thank you for the invitation. This is a community I've gotten to know over many years, directly, very close up with folks like Diego, but also Federico, a whole crew. I also want to give thanks to Rafael Palacios for creating the program that has led to so many great scientists coming out of this community. So let's begin. Let me share screen. And okay, here we are. So today I'm going to talk about some new tools for the analysis and visualization of geographic structure and population genetic data. Before I begin, I want to thank my group. A recent photo from the group shown up top here. But some of the work that's been done here came out of a hackathon from several years ago, where you can see a shot of us there doing this hackathon. The work I'm going to present is based on the GGV browser. That was work done by Joe Marcus, a former programmer, then PhD student, and now graduate of the lab, and Daniel Cook, who is a programmer for the lab. The GeoVar plots I'm going to talk about are work that started in this hackathon and then got developed more in the hands of Arjun Vedanda and Dan Rice, a graduate student and postdoc, respectively. We build on the 1,000 genomes data and including the recent sequence version of it from the New York Genome Center, and we are extremely thankful for those data sets and their generation. So a key topic of interest of mine is geography and ecology and evolution, broadly speaking, across organisms. I tend to do most of my work in humans, leveraging large-scale human data sets, but I come from an evolutionary biology background. There are a lot of questions where genetics and geography, ecology, and evolution all intertwine. And so in population ecology, we're often interested in asking, how are populations connected by dispersal? And how does this affect their long-term demographic trajectories? For instance, populations on either side of the Sierra Madre, are they on independent ecological trajectories in terms of their population sizes and the way that they fluctuate? Are they on independent trajectories in terms of their evolution, in terms of allopatry or being more loosely coupled in parapatry? In conservation biology, we're often interested in asking, what are the geographical groupings that we should manage for conservation? And in complex trait biology, including disease mapping, complex diseases, we often need to understand geography and genetics to help avoid confounding environmental factors with genetic ones as we map traits. And then also as we just design studies to try to, especially in human biomedicine, trying to discover disease variants and understand how to use them for public health. So in all of these areas, it's very useful to understand the population structure of a species, and one of the ways we can do that is with genetics. The big challenge with genetic data is that it comes in these days often in terms of short reads or in terms of probe intensities, low level data coming off of sequencing machines, array genotyping machines, and it's quite a gap between the way the data comes in and the types of biological insights that we'd like to gain. For instance, learning about human migration patterns over the globe. So to make that link, we need to use theoretical models and statistical inference tools and visualization tools to help bridge this gap. And I see this slide as sort of the major challenge that my lab tries to focus on. Today we're going to talk about a challenge in understanding population structure. I'm going to present a browser that we've developed for human genetic variation, then what we call a variant-centric view of human population structure, and then I'll conclude with some discussion. So the challenge that I want us to think about is how there are many different ways of visualizing population structure that we use in population genetics. There are a bunch of examples on the bottom half of the slide. This is a structure plot, the type of plot that comes out of the program structure or admixture or some of the alternatives that fit these structure-like models, PCA plots, tree-like representations of population structure. This is an output from tree mix, and then migration maps. This is an output of the EAMS software that my lab, along with Matthew Stevens, has been developing over many years now. So there are many ways to represent population structure, but all of them have a challenging issue, which is that they don't convey absolute levels of differentiation very well. So you could have the same structure plot for populations that are deeply diverged from one another, or ones that are shallowly diverged from one another, and it's not indicated. You would need to look at some other aspect of the output than this type of visualization to understand that. In PCA, it's not directly represented. It's somewhat contained in the percents of variation explained, but that's more indirect. EAMS, the highs and lows here don't indicate where the levels of absolute differentiation. And in tree mix-like representations, the branch lengths tell you something about absolute differentiation, but it's very difficult to interpret these drift parameters. So for all these visualizations that we use to help us understand population structure in humans and in other species, they actually don't convey one of the most essential aspects about population structure, which is just what are the absolute levels of differentiation? Or put another way, what are typical allele frequency patterns? After looking at one of these plots, can we say what a typical allele frequency looks like? I'd suggest that we can't, and that for many people, even folks who've studied, say, human genetics quite a bit, just on the basis of these, or at least at a low level, in terms of just looking at these plots, they would be hard-pressed to say, well, yeah, a typical variant is going to be 20% frequency, this much over the world, right? So we want to try to address this challenge. One of the standard approaches that population geneticists use to convey absolute patterns of differentiation is FST. So we'll say calculate FST, which is a reduction in heterozygosity relative to the expectations under Hardy-Weinberg, and is one of the key aspects of it. It's measuring the Volland effect. So if these lines represent Hardy-Weinberg expectations, we take global population data from humans, we'll see that there's a deficiency of heterozygotes of about 10% or so, and that's a measure of how differentiated allele frequencies are in humans. If we look within like just a data set from Europe, we'll get much lower values of 0.3%. But I'd still challenge it, even though if we use FST to try to talk about differentiation, it still doesn't imply how a typical variant is distributed. If I tell you FST is 10%, how good are you at saying, well, that variant will vary this much over the globe? So that takes some experience. It takes some extra sources of data. So let's try to get at this question. What are some typical geographic patterns for human genetic variants? That's the challenge. And I like this quote from a paper by Mike Eisen and colleagues from the early days of genotyping arrays, where they recognized that the technology had gotten so large that the main challenge and product of their paper was to address this quote. We need to develop our ability to see the information in the massive tables of quantitative measurements that these approaches produce. Right? Data visualization and genomics go hand in hand from the very beginning. So here we have a browser that we've developed called the Geography of Genetic Variants browser. It's very straightforward in some ways, in that what we're showing is the allele frequencies for particular SNPs. This is a position on chromosome 1, the T and G alleles, their frequencies shown in these pie charts. It gets a little more interesting in that for very rare variants, we're going to change the scale that we work out of. So because like a very rare variant would be a very thin pie slice, we're going to occasionally change the scale to measure out of a smaller fraction. So this is 25% of 1%, or let me show here, 25% of 1%, so it's 0.25%, okay? This variant then is at about 0.5% in just one population in Europe from the 1,000 genomes data, okay? So one feature that we put on this browser, and I encourage other browser developers to do this more, is to put a random button. This is kind of inspired by Google. Take me to a random variant in the genome. And hitting this button several times will help you see what are just like sort of typical things that appear in the human genome. And so what I've done here is create a little video where I've clicked that random button about 15 times or so, and we're going to see different variants flash across the screen. So this is a variant that's at 0.5% frequency in a population in Southeast Asia, and you can, if the colors are going to help reflect when the scale is changing. Red is this 0.01% scale. Blue is going to be the normal out of 100%. Okay, so let's get started. Okay, so that's 0.5% frequency. That's a variant that's about frequency in East Africa, about 0.5% frequency in Europe, Asian population 0.5%, variant distributed across the whole globe. Variant found about 5% in population Africa and a little bit in the Americas. Okay, 0.5% Japan and Asian population, 0.5%, 1%, 0.5%, 0.5% localized. Another variant Americas and 0.5%, 0.5%, Southeast Asia. Okay, so what we saw here were what were the general trends that we saw? We saw lots of variants that were extremely rare and found in just one or two populations, and then we only saw one common variant that was common in any of the populations, and it was found across the whole globe. And then we saw some intermediate frequency ones that were sort of two that were sort of, or three that maybe were shared between some African populations and some of the populations in the Americas. And note that these populations in the Americas are of admixed ancestry. So, okay, so how can we, that's a snapshot of 20. It gives us some sense. There must be a lot of rare localized variants in the human genome. But, we'd like to get a sense of this for all the variants. And and actually, so I wrote 80 million here, but actually we use the New York Genome Center's new version of the data, which is deeper coverage, and the number actually grows of total variants is actually just over 90 million variants. So it's too many to look at. So what we've done is come up with this idea of compressing the information to a basic, what we call GeoVar code. Okay, this is very simple-minded. The idea is that this is simple enough that it can be used in teaching. So the allele frequency map, where like this pattern represents something that is globally common, can be represented by just a 5-digit string, where we're going to use the supergroup kind of designations in kind of an idea taken from the 1,000 Genomes paper, and mark whether a variant is common, rare, or unobserved within a particular region. And so here we have this variant is common everywhere. And then here is a variant that's rare in South Asia. We'll have the code UURUU. So in just 5 digits, we can convey a geographic pattern across the globe instead of having 26 real valued numbers. Let's see, I heard there's a question, so let me, I think it might be hard for me to find the question. So I'll say if it's a clarifying question, maybe Diego, feel free to interrupt me and ask the question, something that'll prevent people from understanding what comes next. But if it's a discussion question, maybe we can take it at the end.
We will take all the questions at the end. Go ahead.
Okay. So, great. Okay. So one note is we use a slightly modified definition of these supergroups than the 1,000 Genomes paper did. We have the populations that were, we have the Ceph Utah populations actually in Utah. We have, and then this has African Americans from the Southwest, Afro-Caribbeans from Barbados, and then the other populations that are in the Americas group, which are of admixed ancestry, such as Mexicans from Los Angeles, Colombians from Lima, the Puerto Ricans, and so on. So, okay, so we will have input these allele counts in the 1,000 genomes data. And for each of them, we can produce this 5-digit code. And now the question is, like, if we understand something about human genetic variation, we should be able to have some idea of what these numbers are, at least what their relative fractions are. How many of this pattern should we see? How many of this pattern should we see? How many of this one and this one? Okay, so you might take a moment and think for yourself, how many of each of those would you expect to see? Okay. And so we're going to count these, and when we can count them, we can sort of order them and get a better understanding of the distribution more clearly. So we're going to ask what is typical, what are the most common patterns of these 5-digit codes to be in the 1,000 genomes data? How many categories will be needed to explain 80%, say, of all the variants? Will we need to use, it's about 243 total categories. So will we need to use all of those categories, or will we need to use a much smaller set? Okay, so let's look at what we see. Okay, so on the left, representation of the whole in the sense that there are 2.9 billion measurable sites in the genome are sort of assayable using sequencing technologies. Of these, 3% of them are single nucleotide variants. And of those, we can break the fraction up by their GeoVar codes. And we see 16% of them are rare in Africa, absent everywhere else. Another 16% rare in East Asia, absent everywhere else. 15% rare in South Asia. Another 13% rare in the Americas. Then we have variants that are rare in Africa and rare in the Americas. We think these might represent variants that arose in often West Africa and then were carried in the slave trade and to the New World, and then rare in Europe, and then variants that are common everywhere. Okay, so the first time we see a variant that's common anywhere, it's common everywhere. Then we see variants that are shared between pairs of populations that, again, like the Africa and the Americas, that we have some sense of recent gene flow. We have Europe and the Americas, Europe and South Asia and the Americas. We have variants that are common in Africa, absent everywhere else except the Americas. I think these are variants that are from the predate the bottleneck and did not leave Africa as humans dispersed outwards. Okay, so there's this very remarkable distribution. We only need 7 out of the 243 categories to explain about 85% of all variants. And so we can that's a kind of very different view of human genetic variation than a PCA plot might get you. We can use this approach to look at other lists of variants. So what I'm going to show on the next slide is the geographic distributions of the variants that are found on genotyping arrays. So these are the arrays that are used to do studies of disease mapping and personal ancestry testing and so on in human genetics. And so you're probing for 2 known alleles at a single nucleotide variant. there are different ascertainment strategies. And so these are 5 different genotyping arrays. And you can see that many of the arrays are highly enriched for common variants, and that the arrays differ slightly in how much variation they cover from different parts of the world. So the omega array having the most sort of rare variants that are in non-European ancestries, because it's the multi-ethnic genome array. It was developed precisely for helping get a broad distribution of variants that are more distributed across the world. Okay, so why is this the distribution that we see over all single nucleotide variants in the 1,000 genomes data? This particular fractionation versus any other, it's the result of the recent African origin of all humans, is what we believe. So, and I'll explain that more in a second here, but what I want you to picture is that you have variants that are, well, let me walk us through this. So this is looking at 2,000 generations in a simulation, and that's about the time scale of the dispersal of humans across the globe. And variants that were common at the beginning of this time period in any like independent sort of daughter replicates would still be common today. Okay. Variants that are rare tend to be lost, and then new mutations that arise since this common ancestry in Africa will tend to be still rare over 2,000 generations. There's very little time. 2,000 generations is too short for any of these new mutations to become common. So what you end up having then are common variants that are old enough that they were found in Africa, and then they stayed common everywhere else, in the spread across the globe. And then you have new mutations that are layered on top of that, but they're so new that they haven't had a chance to spread out and be shared across different regions. You start to see some of it in like the rare here, the rare there, some of the sharing of rare variants. But you have this overall pattern of rare localized variants that are new mutations that follow the divergence, and then common variants that are old enough they're shared across all populations. Okay. Dan Rice, a postdoc in the group, looked at this more formally in theoretical models with 2 populations with divergence and admixture. And if you have a deep divergence on the scale of the divergence time in generations being about half of the pairwise coalescent time, or 2n, you would see a large fraction of variants that are common in one population and absent in the other. But with humans, we don't see that. We see patterns like this, where the most abundant patterns are rare in one population, absent in the other. And when you see common variants, they tend to be shared. Okay. So more consistent with these recent divergence, recent admixture models. Okay. We can now turn and use the same approach to look at the variants that arise when we compare 2 individuals' genomes, individual A versus individual B, and line up their genomes and ask, where are there single nucleotide variants that exist between the 2 genomes, called these pairwise single nucleotide variants, okay? And look at their geographic distributions. And what's interesting here is that even though we just learned the most abundant sort of typical pattern for a human variant is to be rare and geographically localized, when we compare 2 individuals, the main pattern we see is that they carry common variants. That's because even though there are many rare variants out there, it's very rare for anybody to carry them. So when 2 individuals differ, they tend to differ by a common variant. Those common variants tend to be old. Because they're old, they were found in Africa and spread out across the globe. So they're the most abundant type of variant found in these pairwise comparisons. Okay, and so we calculate the proportion of globally widespread alleles. This is like more inclusive than just the CCCC category, the common everywhere. This just means that it's present everywhere. That it's like all the categories where there's no U's, And in all these pairwise comparisons of individuals, it's over 50% of the variance that you find comparing two individuals could be found anywhere across the globe. They could differ between any two individuals in the world. So this is a way of us seeing more directly sort of the low levels and kind of in a more exact sense of the structure that exists in human populations. Across these pairs, it's kind of interesting to try to, these are different individuals from different populations across the globe. And it's sort of an interesting task to try to guess what kinds of pairs are here. These are the exact pairings. With comparisons, like for instance, between a Yoruban and French individual, Yoruba Han, Yoruba, French Han, French, and Han. Okay, throughout you see this shared existence of a high number of globally widespread alleles in all these pairwise comparisons. Okay, we can count, as I said, so we're back to the answer with these four patterns, 13.7 million that have this rare variant pattern, 3.6 million that have this pattern. This particular pattern here, common in Africa, rare in Europe, common in Asia, there's only 8 of them in the whole genome. This is probably some kind of multiple mutation event. This pattern, common in South Asia and in the Americas, absent everywhere else, none of them. Okay, so the patterns that we're seeing, we think are largely the result of this demographic spread out across the planet. As a reminder, we will see outliers from those general patterns, places where there's high FST, high levels of differentiation, where there are variants that are common in one part of the world and absent in the other. And this is a figure from a teaching lab that I use, where I have the students calculate FST in a moving window across chromosome 15. They find this strong peak, and I have them look underneath it. And what they find and when they look in the genome browser is SLC24A5, one of the major known skin pigmentation loci. So all this is to say that the things that we see superficially in terms of how we differ by skin pigmentation are outliers in the genome to this broader story of our recent shared ancestry in Africa. This is a less granular view, sort of breaking it up by 26 populations. And so here I've done SNPs as columns, and these are just for 100 SNPs, and each SNP is a column, and the SNPs are ordered by the global frequency. So we see the bulk of this are rare variants that are geographically localized, and then as we move towards some more common variants, they tend to all be globally shared. And then there's sort of an intermediate transition zone where the variants are kind of shared between several populations that are closer to each other genetically. Okay. This is the view when we do GeoVar codes 26-way, and we see again this sort of like in this level, all these rare variants are geographically localized. Common variants that are globally shared are a huge part of our variation when we're not looking at these young, new, rare localized variants. Okay, so what we've produced is this variant-centric view of population structure. It reveals structure of frequency variation more directly, we think. It reaffirms work of many previous studies. It's not that these are new insights about human genetic variation. We just think this is a useful way to look at it and an important thing to be teaching about right now. In particular, because of the growing use of genetic ancestry testing and PCA structure plots, where there are a lot of misconceptions that can arise, where people can look at a PCA and structure plot and think that there must be deep divergence between the populations. This isn't just a problem in human population genetics. In thinking about conservation biology, just because things cluster in a structure plot doesn't mean that they're deeply diverged and evolutionarily significant units in some sense. So that's one misconception it can address. Another is that one might suppose that because ancestry tests work, that there must be variants that are partitioned into regions of very high and very low frequency, and that's the typical pattern of human genetic variation. And as these results show, that's not the case. And then the idea that most individuals must differ due to variants that would be like local to their population and not found in the other. That would be a kind of natural thing that someone might suppose, but that is also a misconception. Most individuals differ due to common variants that are globally widespread, as we just showed. So this work can also help calibrate expectations in terms of understanding that common variants found in GWAS studies will often be shared, rare variants less so. So it gives insights into how we might think about study design and the spatial scale of our samples, and that's actually another topic that we're working on with my postdoc Dan Rice. And then some final details and caveats before I finish. One is that it's interesting. Archaic introgression is not so apparent here. It's not that you see a pattern that is due to Neanderthal or Denisovan introgression. That's partly because there's a small fraction of the human genome that is the result of archaic introgression. Also, because introgression was old, the allele frequencies have spread out and cover many. They now don't kind of cluster into one of these patterns. Should be said there's uncertainty in these category assignments because of sampling error. So the U category is likely overestimated because rare variants are going to be missed. That said, we have taken some approaches to account for frequency uncertainty and improve the frequency estimation using empirical Bayes approaches, and the global pictures that I show only shift slightly. So I present this very simple approach because it's easier to explain and maybe more transparent for teaching and convincing people about human variation. The 1,000 Genomes Project had certain criteria for inclusion in terms of certain samples having, you had to have 3 grandparents or greater from the local population to be included. So, the true local diversity in any one of these places around the world is going to be greater than what's depicted. And then, for some applications, this CCCC category, which is it's encompassing many variants, has meaningful variation. So, a lot of the early structure plots and so on are done with common variants on arrays that are largely this type of variant. For applying this in other species and settings, we think it could be useful for it being a new form of visualization. But one has to be careful that the units are sampled about equally, because you'll have a differential power to detect rare variants if you have much larger sample sizes in one group than another. So subsampling may be necessary to achieve more equivalent sample sizes, or there's a sort of project to be done, future work in terms of trying to incorporate uneven sample sizes more directly in the method with some corrections. And then finally, that 5% threshold is one that we use because it's sort of a typical threshold used in human population genetics. It tends to it actually, nicely, and sort of, natural human genetics, but in other settings, other thresholds may be sensible. Okay, so before I finish, I just want to zoom out, and I love this plot that came out when I was a graduate student. Some of the earliest sequencing of the X chromosome across many different primates, and it helps put human genetic variation in perspective of our primate neighbors and how closely related all of our samples are. So this is another example, I think, of an effective visualization. I think these tools of visualization are something I'm investing time in right now because it's, I think, really important that we are helping broad audiences understand genetic variation, and especially in the issues between genetics and racism that are really still a major problem in our world. And I appreciated the conversations that took place yesterday around this in the Mexpopgen meeting, the sensitivity to these ethical and societal issues which our field touches on. And I want to give a shout out to Michal Sohail, who's been a postdoc in my group, has been helping me a lot in thinking about these issues. And we've been running a reading group at UChicago around this. So let me finish. I did the acknowledgements at the beginning because I knew I might run out of time. To say that there's funding for a postdoctoral position here. And with that, I'll stop. Thanks so much.
All right. Thank you for that, John. We are running out of time, so I will only have time for maybe just one quick question. And there's an interest from members of the audience, particularly Alicia Mastreta and Consuelo Daisu Quinto, about if you can use the browser for genetic variants in other species, or if users can upload their own data.
Yeah. So right now we it would be possible to reconfigure it and sort of run a local one. The one that we're serving up right now is, we just have several human data sets, but the code for the browser is something that can be shared, and people can start up like sort of local instances with their own data. That's something we need to kind of help make like more manuals and documentation for, but I think that's a great idea.
That sounds good. Well, we are running out of time, so I'm sorry with members of the audience, but we won't be able to take their questions because we need to move to the next session. But thanks a lot for your talk, John. This was very interesting.
Great, thanks. My apologies, and I'll be at the tables to talk more afterwards. Thanks.
That sounds good.
So just to complement what Dave was saying, now we have a small break, so everyone can go to the virtual tables and talk to someone else, among them John, who has a lot of questions by our audience. So yeah, we'll start the next session, 9:45.
Great.